Girls, today in this video I will explain a little bit on this experiment that is on the preparation of the standard curve for amino acids using Ninhydrin. So this amino acid I had already discussed with you in the theory. So if you remember that there are 22 different standard amino acids, okay, which are commonly found in proteins. Okay, there are around 22 standard amino acids, and also you have seen when we study the chemical properties of amino acid, we know that Ninhydrin, which is this compound, this Ninhydrin reagent, it reacts with amino acid, and as soon as it reacts with amino acid, it turns deep purple in color. Okay, so a deep purple is this deep purple color that is detected in this method. So Ninhydrin, it usually react with 3 amino group of the amino acid, but it does not react with the proteins or the peptides. Okay, not with the proteins or the peptides, but with 3 amino acids. So you see, since you see here with a free alpha amino group, and we know that this group is present in all the amino acids, proteins or peptides. So if you remember, when we study the chemical properties of amino acids, they react with Ninhydrin to form a blue colored compound known as the Reagent's purple. Okay, known as the Reagent's purple, and it is this purple color. Through this purple color, we can detect the presence of amino acids in a given sample. Okay, so here in this experiment today, we are not only going to prepare a standard curve, but we will also find the unknown amino acid concentration. From the standard curve, we will also find the unknown. So this method, okay, for detection of amino acid using Ninhydrin, for preparing the standard curve using Ninhydrin, is one of the best method for detecting amino acids. Okay, and this particular experiment, okay, it has been used conventionally to detect the microgram amounts when amino acid, okay, the microgram amount of amino acid, okay, to detect the amino acid using this Ninhydrin, this method. And when amino acid with a free alpha amino group, they are treated with excess of Ninhydrin, they yield a purple colored product. And under appropriate conditions, the color intensity produced is proportional to the amino acid concentration. That is why I told you that since we are giving a different amino acid concentrations here, so you can see the intensity of the purple color also in all the test tubes are different. Some are more darker, some are lighter in color because of the less concentration of amino acid, and those which are more dark in color, it's because the concentration of amino acid is high. And here the primary amino groups, they react with Ninhydrin to form the purple color dye, which is known as the Reagent's purple, and this was covered by Siegfried and Riemann in the year 1910. Okay, so this we had already discussed in the theory also. So just remember that the principle behind this, okay, is that the Ninhydrin, it will react with the free amino acid. The decarboxylation reaction, it will proceed for free amino acid, and it will not happen for peptides and proteins. So Ninhydrin, it will react only with the free amino group of the amino acid. And this is one of the best method for detecting the amino acid in a sample. Is one of the most important method. So first of all, in this experiment, see what are the things which are required. Now we need the standard amino acid, then we need the buffer, then the Ninhydrin reagent, ethanol as well as distilled water. Okay, so the chemicals that we are going, the reagents that we are going to use in this experiment, I've already prepared. Now the first reagent that we require is sodium acetate. Okay, the sodium acetate buffer. So for preparing a sodium acetate buffer of 0.2 molar and pH 5. Okay, so the buffer, it has to be at pH 5. Now for the buffer, in order to prepare the buffer, we require this sodium acetate, and we require this glacial acetic acid. We will mix this together. Okay, the glacial acetic acid, we have to prepare 12 ml of this in and 54.43 gram of this in 2 liter of water. So after mixing these two, we need to measure the pH of this buffer. It should be at pH 5. So for that we need to use a pH meter, and we also need to prepare this sodium hydroxide. Okay, why? Because this is this acetic acid, we know that it is acidic, so it will be, the pH will be below 5. So in order to make the pH to 5, we will use this sodium hydroxide. We will prepare this in 10 normal. So I have already prepared that. So after we get the pH of this buffer at 5, then the next reagent that we require in this experiment is the Ninhydrin reagent. Now for Ninhydrin reagent, first we need to prepare 8% Ninhydrin. Okay, so to prepare this 8% Ninhydrin, we need to prepare this 8 gram of Ninhydrin in 100 ml of acetone. Okay, we will dissolve this Ninhydrin in acetone, and later on the Ninhydrin reagent we will prepare by taking this sodium acetate buffer. Okay, we will take this sodium acetate buffer 92 ml. We will mix with this Ninhydrin 8 ml. Then we will have this Ninhydrin reagent. So this reagent we will use in our experiment. And the last reagent that we require here is the standard amino acid. So this standard amino acid also I have already prepared. Its strength is 200 microgram per ml. Okay, so it is 200 microgram per ml, the strength of this standard amino acid. So girls, this is another instrument that we are using in this experiment. This is the pH meter. Okay, so for measuring the pH of the buffer, we will use this instrument. Now before using this pH meter, we need to calibrate. Okay, so we need to correlate the pH of this instrument with the standard pH. So we will use water sample here. Okay, we will use water sample in order to correlate it with the instrument. We will make that, because we know that the pH of the water is 7, so we will calibrate this pH meter at pH 7. So first we need to calibrate. So I am taking distilled water here. So first of all we will calibrate this instrument. From here you have to calibrate to set the pH to 7. Okay, that means the neutral pH. We know that water, it has a neutral pH, neither acidic nor basic. So I have already, you see here it's 7, the pH is 7. So I have already calibrated the pH of this instrument at 7. I have set it at 7. So now we will measure the pH of this buffer. If it's less than 5, if it's more acidic, because you see that the glacial, we add glacial acetic acid right in this buffer, so it is acidic. So if it is too acidic, we need to bring the pH to 5. First, let us just see the pH of the buffer is how much. Okay, so for measuring the pH of the buffer, we need, you see here it's 6.9. So if it's more, we need to bring it down. You have to add acid. And if it's less, suppose if it's 5, if it's 4 or 3, that means below 5. So in order to bring it up, we will add, in order to make it more basic, okay, so we have to add, we will add sodium hydroxide that we have already prepared in order to make it acidic, more acidic. So in order to bring down this one, now we need to add acid. So in this way, you have to use this instrument to measure the pH of this buffer. So first of all, what we are going to do, I will tell you here. You see in the test tube. So first of all, you will take blank test tubes. Okay, first of all, you will take blank test tubes. Then we will label this test tube as the first one as the blank. Then we have 1, 2, 3, 4, 5 different concentration of amino acid. Okay, different concentration of standard amino acids will add in this test tube. So now what you will do, you will pipette out a different concentration of amino acid. So since this is a blank, so here we will not add any amino acid. Then in the first one, we will take 0.1 ml. Okay, 0.1. Then we have, we'll take 0.2, 0.4 ml, 0.6 ml, 0.8 ml, and 1 ml of amino acid. And actually we need one more test tube where this is the unknown amino acid. Okay, so we will take one more test tube, and here we will add amino acid, and it is unknown. Okay, you don't know the concentration of this amino acid. So now next what we are going to do is that here, now after taking the different concentration of the amino acids, so we will make the volume of all these to 4, okay, the final volume to 4 ml by adding water. So here, since this is a blank, there is no standard amino acid here, I will just add 4 ml water. Then in the first test tube, there is 0.2 ml amino acid. So I will add 3.8 ml water to make the final volume to 4. Then same thing here in the third test tube, we have 0.4 ml amino acid. So I'm going to add 3.6 volume of water in order to make the final volume to 4. So in that way, we are going to add distilled water to make the final volume to 4. Then the next step is you will add the Ninhydrin solution to the, you will add Ninhydrin 1 ml Ninhydrin solution to all these test tubes. Okay, and the last step, after adding Ninhydrin solution, what we are going to do is we are going to keep the sample for 15 minutes in a boiling hot water bath. Okay, so this is a water bath. Okay, so we will on this water bath and we will keep the test tube inside. Then the reaction will occur. Then after 15 minutes, after taking out these test tubes, we will add the we will add the ethanol to stop the reaction, and then we will be taking the reading using a calorimeter instrument. Okay, this is the calorimeter. Okay, it is an instrument where we use to take the absorbance. So depending, if you see here, depending on the absorbance required, our experiment, okay, we require this 570 nanometer absorbance. So we will make this, we will keep this filter at 7. Here you can see there is a filter here. So we'll keep it at 7 because we need this particular wavelength, okay, 570. So, sorry. So you can see after taking out these test tubes from the hot water bath, the color it changes to purple. Okay, at first you will see it is like this, like the blank only, but after the reaction occur, because we have already added this Ninhydrin reagent 1 ml to each test tube. So as you can see that the blank, it does not change color. Why? Because here only we have added only water. So there is no amino acid, so no reaction takes place. Whereas in all these test tubes, because we have given different concentrations of amino acids, so the color also you can see it is slightly different from for each test tube. If you see, the color is different because we have given different concentrations of amino acids. Okay, so always when you keep this inside the water bath for 15 minutes, we also have to remember that we have to cover this test tube with the aluminum foil. Then only we will keep in the water bath. We'll let the reaction to occur. After taking out from the water bath, these test tube, you will let them to cool down, okay, or you will keep it inside the cold water. And after that, the last step, as I said, is we will add ethanol in order to stop the reaction. So now we will take a reading for these. We will take the absorbance reading. I will show you in this calorimeter how to take the readings. So you can see that you have to note down all the readings, all the absorbance readings of these samples. Okay, so after taking the absorbance readings of these samples, then we will plot a graph. So for taking the OD reading, okay, the absorbance reading, now this is the calorimeter tube. So we will pour first the blank. Okay, we will pour this blank in this calorimeter tube, and then we are going to use this calorimeter instrument. Now this is the part where you have to keep your tube, your calorimeter tube, and then we have to cover it to prevent any light to enter. Then for first of all, you have to set this calorimeter, you have to set 0 with the blank. Okay, so I've already kept the blank sample inside. Then now I will set 0. Okay, after setting it to 0, after you set this to 0 with the blank, okay, using the blank sample. Now I will take out the blank. Then in the same way, you will measure the reading for all these other samples. Okay, you will measure the absorbance reading for all these samples using this calorimeter. So now taking the first sample, okay, I will take the reading of the first sample. Let us see. So you can take the reading from here of the first sample. Okay, so in this way you will take the reading of all the sample, and then we will plot a graph for the standard curve. So after plotting a graph for different concentrations, now this is the standard curve that we are getting. Of course, there are some slight errors here and there, but this is the standard curve. Now from the standard curve, we will since I have given you the unknown also in this experiment, so the unknown here that we are getting the absorbance reading is around 0.65. So I will take this 0.65, then I will connect this one to the standard curve, and I will drop down in the x-axis to see the volume. Okay, to see the volume of that standard of amino acid in the unknown. See, when you plot a graph, you have to plot the volume of the standard amino acid against the absorbance reading, okay, against this OD. So this will be on the x-axis and the OD will be on the y-axis. So after getting the standard curve, we found out that from the graph, the volume of the unknown is 0.46. Okay, so 0.46. Now, since the standard amino acid, I've already mentioned that the standard amino acid that we are doing here in this experiment, it is 200 microgram per ml. So in this experiment, they are doing 150 microgram, but we are doing 200 microgram. So how you will calculate? See, from the graph, it was observed that in this absorbance reading, it is equal to 0.46 ml of amino acid, right? Just now from the graph, you can see that it is equal to 0.46 ml of amino acid. So since our standard amino acid that we are taking in this test tube, it is 1 ml of standard amino acid contains 200 microgram of amino acids. So therefore 0.46 ml of the unknown amino acid solution, it contains 69. How we are getting this? Since 1 ml is 200 microgram, so 0.46, it will be 0.46 into 200 microgram. Okay, so like that you can find out the the concentration of the unknown amino acid from the standard curve.